Hello students, in this video we will discuss about the antibody and its types. An antibody, also known as immunoglobulin, is a large Y-shaped protein used by the immune system to identify and neutralize foreign objects such as pathogenic bacteria and viruses. The antibody recognize a unique molecule of the pathogen called an antigen and are specialized protective proteins that binds like a lock and key to the body's foreign invaders, whether they are viruses, bacteria, fungi, or parasites. When an antigen binds to the B cell surface, it stimulates the B cells to divide and mature into a group of identical cells called a clone. The mature B cells, called plasma cells, secrete millions of antibodies into the bloodstream and lymphatic system. Now let's talk about the antibody types. Antibodies are grouped into five classes or types according to their constant region and inactivity. The five primary classes of immunoglobulins are IgG, IgM, IgE, IgA, and IgD. IgG have gamma chains, IgM have mu chains, IgE have epsilon chains, IgA have alpha chains, and IgD have delta chains. Now let's discuss about the IgG. All IgGs are monomers and has two heavy chains and two light chains of either K or lambda types but not both. Fc region has CH1, CH2 and CH3 domains. IgG is 7S immunoglobulin with molecular weight of 160 kDa and it is found highest concentration in blood. Subclasses of IgG in humans are IgG1, which is 65 to 70%, IgG2, which is 23 to 28%, IgG3, which is 4 to 7%, and IgG4, which is 3 to 4%. Second type is IgM. Normally exists as a pentamer and 19 as immunoglobulin and also exists as a monomer. On B cell surface, IgM is single monomer, but the secreted IgM is a pentamer. Five monomers of IgM are linked in a circular fashion by disulfide bonds between heavy chains and one addition chain rich in cysteine and asparagine called J chains. In the pentameric form, all heavy chains are identical and all light chains too are identical. Thus, the valence is theoretically 10. Each IgM monomer has two heavy chains and two light chains of either K or lambda. Each heavy chain has an additional domain at the C terminus of Fc region. IgM do not have hinge region. IgM is the first immunoglobulin to be made by the fetus or the first immunoglobulin to be made by a virgin B cells when it is stimulated by an antigen. Now IgE. IgE exists as a monomer and has an extra domain in the constant region. It mediates immediate hypersensitivity reaction, for example, anaphylactic shock, hay fever, asthma, and etc. IgE is the least common serum Ig since it binds very tightly to Fc receptors on basophils and mast cells even before interacting with antigen. It is involved in allergic reaction and also play a role in parasitic Helminth diseases. Since serum IgE levels rise in parasitic diseases, measuring IgE levels is helpful in diagnosing parasitic infection and IgE does not fix complement. Fourth antibody type is IgA. IgA occurs in two forms, which is serum IgA and secretory IgA. The serum IgA is a monomer which is 7S with molecular weight 160 kDa, but secretory IgA usually found in mucosal surfaces and in secretion is a dimer. It is found by 2,7S IgA monomer joined at the carboxyl terminus of Fc region by J chain and also with a secretory component. The dimer IgA is much longer, which is 11S with molecular weight of 360 kDa. IgA molecule has two alpha heavy chains containing three constant domains, which is CH1, CH2 and CH3, and either 2K or 2 lambda light chains. IgA, since it is found in secretions, secretory IgA is important in local, like for example mucosal immunity. Normally, IgA does not fix complement unless aggregated. IgA can binding to some cells, which is PMNs, and some lymphocyte and helps in phagocytosis and intracellular killing of microorganisms. It is a minor component in systemic humoral immunity but plays a major role in mucosal immunity and found in gut contents or feces are known as coproantibodies. The last type of antibody is IgD. IgD exists as a only monomer and is found in low levels in serum, and it play role in serum uncertain. It is primarily found on B cell surfaces where it functions as a receptor for antigen, and IgD on the surface of B cells has extra amino acids at C terminal and for encoding to the membrane and does not bind complement.